So I'm Yamagishi, and I'm going to introduce you about our experiment going on the International Space Station. And you will know, or you will see what Tanpopo means in a few minutes. And we have two scientific targets of Tanpopo experiments. One is chemical revolution. We have learned a lot in yesterday's session. There are many molecules of organic compounds found in molecular cloud. And in asteroids and comets, there are organic compounds. And they must have accumulated, the organic compounds must have accumulated on the surface of Earth before the origins of life. But one of the remaining issues is the amount of the organic compounds accumulated, because we don't see many asteroids or comets coming to the Earth. But one of the candidates is the cosmic dust. The cosmic dust, smaller than 1 millimeter, can softly landed on the atmosphere of the Earth and to the ground of the Earth. Even now, we have more than tens of thousands tons of cosmic dust are arriving on the Earth per year. So our target is to test if cosmic dust can contribute to the origin of life or not. And the second target is the panspermia hypothesis. And most of you know well that pan means everywhere or all, and spermia means spores or the seeds. So more than 100 years ago, Arrhenius proposed that the life might be everywhere in the space and can be transported from Earth to the other planet or the otherwise. To address these questions, we are doing a couple of experiments, sub-themes going on. To address the panspermia hypothesis, we are doing the capturing experiment of microbes on the International Space Station. And we are also exposing the terrestrial microbes on the International Space Station. To address the second theme, chemical revolution, we are trying to capture the organic compounds included in the cosmic dust. And we are trying to test the survivability of the organic compounds exposed in space. In addition to these two scientific targets, we are also trying to test the availability or the usefulness of the aerogel as a media to catch the hypervelocity impact microparticles. And the other is to monitor the space debris for the safety of the space mission. And this is Tanpopo. I think you have seen it yesterday. They were anywhere. And the seeds of Tanpopo means dandelion in English, and the seeds of dandelion are spread by wind. The process resembles the panspermia hypothesis. That's why we named this mission Tanpopo. The exposure experiment started on May 2015 on ISS. And this is ISS. It is about 100 meters wide, about 70 meters long. This is orbiting around the Earth about 400 kilometers above surface and every 90 minutes. And the ISS is controlled to direct one side always at the space station. And then these areas are for the astronauts to do some experiment inside of the cabin. And there is outside is for the exposure experiment. And if you expand this area, the JAXA has developed the exposed experiment handrail attachment mechanism. We call it EXHAM. And they were attached to the side, the front side of the exposure facility of GEM of ISS. And we have employing the two types of the experimental instrument. One is, we call it capture panel. A capture panel contains a soft material, we call it aerogel. I'm going to explain it in the next slide. And the other one is exposure panel. The exposure panel is 10 centimeter per centimeter square and composed of 20 units. And each unit contains one of the microbes or the organic compounds for exposure experiments. And this is a capture panel, and which contains two-layered aerogel. And aerogel is a porous silica gel to capture hypervelocity particles with low damage. And we have developed a very low density aerogel, which is 10 milligram per cubic centimeter. This is one of the lowest density aerogel ever built and used in space. And because it is very fragile, we have to support this from outside using a little bit harder aerogel containing 30 milligram per cubic centimeter. And very low density silica gel was designed and used to capture hypervelocity particles with low damage. And this is the experimental schedule of our procedure. And we have launched all of the apparatus needed for our experiments on May 2015. And 3 of the exposure panels were deployed at the same time. And then after 1 year, we have detached one of it and returned to the ground on October 2016. And the second one was exposed for 2 years and detached at July 2017 this year, and it returned to the September of this year. And the third panel is going to expose for 3 years and will return next year. And among the 33 capture panels, and for the first year, we have used 12 of it. And then they are detached and returned to the ground last year. And the second set consists of 12 aerogel panels, was returned this year. And we are now exposing the third set at the ISS. And to review a little bit, very short review of the exposure experiment is shown here. And there are several experiments already done, mainly by the European groups and Russian groups. And if we summarize them, and spores of Bacillus saccharis in monolayer were killed by solar UV. And however, the Bacillus spores in multilayer survived for 6 years in low Earth orbit. And also the rock colonizing lichen, Xanthia elegans, survived for 1.5 years in International Space Station orbit. So, learning from these previous results, we have designed exposure experiment to check the density or the thickness necessary to survive in space, and we are designed to test the survival curve depending on the time. Based on the previous result, European group has proposed the litho panspermia hypothesis. Litho stands for the rocky, and so rocky panspermia. And the previous results suggested that the small rock could be sufficiently large to protect microbes against the intense solar UV. And we are not trying to argue against this hypothesis. Instead, we are trying to another possible hypothesis. We are going to test another additional hypothesis. And we named it massa panspermia. Massa means aggregate. So based on the ground experiment we have done, so we have proposed that the sub-millimeter cell aggregate can survive in space for a long time. Because we know that microbes sometimes has a tendency to make aggregates. They are often called microbiofilm or biofilm. So the dried microfilm can be the way of transferring through the space. So to test this, in the exposure panel, we see the cross-section of one of the unit. There are 2 aluminum plates placed at the upper aluminum plate is illuminated by solar UV, and the lower second aluminum plate is used as a dark control, shielded from UV light, but both plates are exposed to the vacuum and also the radiation. And then in the aluminum plate, we hold the hole, and then in the hole, we place the microbes with different thickness, ranging from 1 micrometer to 1.5 millimeter, to test what type or how deep is necessary for the microbes to survive. And this is microbes we have used. And one of the microbes is called D.radiodurans. This is a very famous microbe known as radio-resistant microbes. Radio means, of course, radiation, and durance means resistance. And the original Deinococcus radiodurans was isolated from canned meat after gamma-ray irradiation for the sterilization. And the other one is Deinococcus aureus. This is our isolated from high altitude. And this is the result. We have tested after 1 year exposure of the microbes. And then the red one is the ground control. We kept the same sample in laboratory. And after 1 year, it showed a very high survival, ranging from 100% to 10% after 1 year, depending on the thickness. And in the ISS cabin, we saw a little bit lower, but a very high survival after 1 year. Comparing with these two controls, the space-exposed samples were dead if the thickness is less than 100 micrometers, illuminated by solar UV. But if you see the thicker samples stayed alive, comparable to the ground or the ISS cabin control. And the similar was seen in the other Deinococcus species, Deinococcus aureus. Comparing with the ground and ISS control, the comparable or a little bit less survival rate was observed when the sample was exposed to the solar UV. But if the sample was not thick enough, and it is 100, they are dead. And we have checked the sample's DNA, and this shows the intactness of one of the genes, ROPA-B gene. Forget about the name, but we checked the intactness of one of the genes. And comparing with the ground control and ISS control, when the thickness is not thick enough, 100 micrometer, and they are exposed to the UV light, most of the DNA, our gene, is destroyed. And that's why we have the dead microbes after 1 year exposure. But interestingly, after even exposed to the right thicknesses more than 100 micrometer, we have only a small amount of the lowering of the intact DNA copy. And that is reasonable because they have the similar survival activity of the microbes. But if you carefully analyze the larger DNA in the genome level, we could detect a little bit different. And for example, this is the fresh cells is prepared, and this band, each white band shows the DNA fragment ranging from 500 genes, approximately, to 100 genes. And each fragment is showing a fragment of the genome level. But even the samples contained or stored in the ground, they are destroyed, and the intensity of the DNA fragment is diminished a little bit. And in the ISS, similar can be seen in this lane. And if the sample is exposed, a severe damage was observed for the genome level. And however, because we have recovered a sufficient survival rate after the exposure of 500 micrometers, so that must be repaired when the sample was re-wet and placed on the surface of the medium, growth medium. So to check what kind of the DNA damage happened and what kind of the DNA repair system is important for the survival in space, we have exposed the DNA repair system mutants. And there are many known damages depending on the space environment. For example, this is the DNA, schematically expressed. DNA consists of two strands of the main strand, and the DNA bases attached to the main strand. And bases are modified or even deleted based on the effect of the space environment. Or sometimes double-stranded are totally broken, cut out. So there are repairing systems to repair, for example, base damage. The system is called nucleotide excision repair system. And if there are double-stranded breaks, those double-stranded breaks can be repaired by either of the homologous recombination system or non-homologous end joining system. So we have used the 3 mutants deficiently, one of these 3 systems. And as I have shown in the previous slide, if the system is totally intact, they can survive for a long time if the thickness is 500 or 1000 micrometer after 1 year. But if the mutants are exposed to the space and comparing with the ground control, the survival rate was significantly lowered, indicating the repair system is important, and the 3 of the important repairing system is important for survival in space, and suggesting the base damage is occurring and double-strand breaks are occurring unless they are repaired afterwards. And in conclusion of these experiments, 5 micrometer is sufficient to protect cells on the surface from UV radiation in space. So that if we have placed the dried cells and expose them to the space environment, 1 micrometer surface cells were dead. But covered by the dead cells, the cells lower than this was protected from the UV and other environmental factors. And double-strand break was induced by radiation, and they are repaired when re-wetted in the medium. And the survival of the microbes has already analyzed for after the 1 year. So we are now continuing the experiment for 2 and 3 years. If we can see the similar survival rate, that suggests the microbes can arrive for a very long time. Instead, we see the continuously dying line, then they cannot survive for a long time. So I'll go very quick for the rest of the time. This is the result of the organic compounds. We have exposed the organic compounds of glycine survived after the exposure, and the simulated interplanetary dust organic compounds survived for long. But hydantoin was disappeared even under the surface. Okay, and this is the initial inspection of the curation of the capture panel. The aerogel was placed in the transparent box, and they were inspected by microscope, and then we found 120 hypervelocity impact larger than 0.1 millimeter. And this is the example. This is the entrance, and you can see some tracks entering downside. And this is another example of the tracks, and there is some particles we can see at the side and the end of the track, impact track. And sometimes we can see totally different shapes of the tracks, and looks like the burst, we call it the burst type. The fragile fractal particle with high velocity may contain organic particles, might make these types of the holes. And some of the tracks were cut out, and this is aerogel cut out. We can see from the side view of the tracks. And this is the crater-shaped track. And then we are now on the analysis of the microbiological analysis and the track analysis to check the structure using the synchrotron radiation we have learned in the morning session. Or the particle is going to be analyzed as SEM-EDS and micro-FTIR, and I'm going to show 2 examples of this line of the analysis. And we have analyzed one particle from the space phase, and that contained the calcium and oxygen and carbon. And the second one at the side phase was composed of silicate and Si and O and sodium. And it is different from the aerogel, which is also consists of Si and oxygen. And by analyzing the microzones, we can see that carbon and calcium and the spectra of these atoms are very similar to calcium carbonate. And we are not sure about the second particle, but maybe similar to glass or the quartz. And micro-FTIR supported these result. And we have also analyzed the isotope of carbon and oxygen. And this is compatible with that these particles may came from the outside of the Earth, but it is not clearly indicated by the isotope analysis. And in conclusion, the Tanpopo mission investigated the panspermia and chemical evolution. And Deinococcus cell aggregate larger than 0.5 millimeters survived for 1 year in space under sunlight. And simulated interplanetary dust organic compounds survived 1 year in space. And 120 hypervelocity impact tracks were found in aerogel blocks, and the analysis are ongoing. And I'd like to thank our members. I have only showed the emblem of each institute or universities. And I'd like to thank JAXA and NASA for the operation. And I'd like to thank all of you. Thank you very much.